So, how to build a model? So, in this case mainly we can use the multivariate analysis, right, to relate the structures and the molecular descriptors with the biological activity. So, here we have the independent variable as molecular descriptors, right, and the dependent variable this is the biological activity, because we need to predict the activity, right. Here this is the dependent ones. So, then what are the other variables? There we can choose, you can change, but the biological activity we cannot change, right, because biological activity obtained from experimental data. So, that is the known values. So, there is a dependent one. So, the independent variables that you can change, how to select the variables and what are the variables which can better represent the biological activity. So, then we can use different types of multivariate analysis to define these different descriptors, right, and how we can link these descriptors to explain the biological activity, right. So, the classical approach, right, as we know, right, is the linear regression technique, right. For example, if we have only one variable, so you can easily do y equal to mx plus c. So, what is y?
Dependent variable.
This is the biological activity. So, this m or this x, this any property, right. So, what is m? So, m is the slope and c is the constant, right. So, if there is only one variable, if you like to add more variables, right, in this case you can develop multiple regression technique y equal to a0 plus a1 x1 plus up to an xn, right. So, for each compound we get one equation, because we know the activity and we know all these properties, but only the coefficients we do not know, right. Then we use the principal of least squares, we try to build this model. So, get the coefficients and then use the coefficients to get the activity of any new compound, that you can easily do with this multiple regression technique, right. Then sometimes the null properties are not straightforward, so you cannot fit with the linear regression. In this case we can also use some kind of nonlinear techniques, right, and you can also use the different types of machine learning. I will discuss later about the different types of machine learning. So, for example, neural networks, support vector machines, right. So, we have the training data here, right, and here this is the machine learning. So, we put in these machine learning techniques. So, this will try to fit the data for all the features plus the activity, and then we will get the data. So, then evaluate the performance, right, then see whether it is fine or not. Otherwise, based on the performance you can give the feedback, so this is the performance obtained with this type of features, then machine learning will optimize, right, could they get the optimal features, so that we can get the best performance, right. We will discuss the machine learning techniques maybe in the later classes, right. So, then what are the various rules of Qsar? For example, if you have 100 data, can we use 50 variables to relate the activity? We cannot do it. So, are there any rules, right? So, yeah, generally we can use the data set contain at least 5 times as many compounds, right, as the descriptors, right. Otherwise, you can fit the data, but it will cause overfitting due to the inclusion of various number of descriptors, and this will work for the training set, but it will fail in the test set, right, or any blind set. So, in this case you have to maintain that the descriptors or the features should be as less as possible compared with the experimental data available that to use for developing the model. Then you need to remove the variables with the 0 variance, and also you have to remove the variables with no unique information. If the variable, sometimes we have some features, there is no information or no relationship with the activities. In this case, we need to remove that variables, because it is not possible to explain why the specific feature is selected to relate the biological activity, right. So, if you look into these Qsar models, in most of the cases if you see the final model contains the most important 3 to 5 descriptors. You can have a large number of descriptors, make a subset and train, right, then again take the best features, make a small subset. Finally, if you end up with the 3 to 5 descriptors, right, eliminating the with high correlation among these properties, then you can see whether a model can explain well with this biological activity. So, there are several rules for the Qsar when you are developing any specific models, right. Then how to evaluate? How to evaluate the Qsar model? So, if you develop a model, right. So, we need to evaluate the performance of the model. So, in this Qsar there are various ways to develop to analyze or evaluate the performance. One is the correlation coefficient, right. This will tell you how far the two variables are related with each other, right. For example, one is x experimental and y is the predicted, right. Then you can use this formula, right, to see whether they are related with each other, right. What is the normally the R value, the range of R values ranges from -1 to +1. If it is -1 they are inversely related, and +1 it is positively related, and if it is 0 it is not related, right. Then depending upon the numbers, for example 0.7 or 0.8, we can say how far the predicted values are related with these experimental values. This will tell you whether the model is correct or not, right, how far you can rely, right, your new model. Then you can see with the root mean square error, right. How to get the root mean square error? Right, you can see the predicted ones, experimental ones. For example, the predicted values are 10.0 and 11.0, and the experimental is 9.1 and it is 9.2, and if this is 12.7 and here it is 11.6, right. So, how to get this root mean square deviation? Calculate there. What is the error for the first one? This is 10 minus 1, right, this is 1 square 1 plus, second one is 0.1, so 0.01, third one 1.1, right, 1.21, right. So, add up 2.22, right, divided by 3, right, this equal to 0.74, right. So, this equal to 0.9, 0.8, yeah, right. So, you can say if this, because this is 1, this is 1.1, right, this is 1.1. So, you can see a balance among all the data which you use in your model. So, from this you can calculate the RMSE, and you can have cutoff values. For example, it is less than 1 or less than 0.5 depending upon your data size or depending upon the values, actual values, right. For example, the active values are around 10, 9, right. If it is 0.8, then how many percentage deviation? 10% or 8% deviation, right, right, yeah. So, you can see, but also in this case we need to be careful, because sometimes these values are less, right, then whatever value you get this deviation, then if you take the percent that will be high. So, in this case we need to use different measures, right, to see whether your predicted values are related with this experimental data, right. So, you can see n is a sample size, so n is a sample size, and here x and y are the predicted experimental values. So, you can get the root mean square error. This will tell you how far your method could fit well with this experimental data. Then how to validate the performance? You can use various ways to validate the performance. Either you can divide the data set into training set and the test set, right. If you have the different data, right, you can use this as training, and this is a test set, right. Then you can use the data in the training to develop your model, and you can use the values to test the new data whether this can be able to predict or not, right. So, you can see these numbers. For example, it can use various percentage, right. For example, you can take 80% here or 20% here or 90-10 or 70-30, right. You can use various proportions and see whether even less number of training you are able to capture the features and whether it is possible to predict, right, the remaining cases with the high reliability, right. This you can do that. So, then we can see then also you can do with the external test, right, and evaluate whether your model works fine or not. This is the commonly used cross validation, this is called the inferred cross validation. In this case we classify the data set into n groups and take the n-1 group as training and the left out for the test. So, in this case if it is the tenfold cross validation, right. So, we can make into 10 groups, or if you have 5-fold cross validation, for example 1, 2, 3, 4, 5, right. So, we use this for training and this for test, and the second case you use this for training. First case is 1, 2, 3, 4 training and 5 as test. The second case you take 2, 3, 4, 5 for training and 1 as test. Likewise, how many times you need to repeat? 5 times you need to repeat and then take the average. This will give you, okay, so how far your method could predict, right. Then we need to resample again, right, and take another 5-fold cross validation. So, the samples will be different, right, and see how can you do that. So, repeat several times and almost all the time we get similar level of performance, then you can see that your method is reliable and you can use your model, right, for predicting any of these compounds, right. Now, I show some case studies I show you. So, here we have the different compounds, right, these are the different 25 compounds, IC50 values are known in micromolar. It is against the epidermal growth factor receptor. This is the well known oncogene, right, and its mutations and overexpression are frequently observed in many of the cancer types. This is very important. So, it is very important to design different types of inhibitors. So, we have the values for the 25 compounds against the EGFR. These are the 25 compounds and these IC50 values in micromolar, right. Now, can we predict these IC50 values using any properties, or if you have the new compounds, because many compounds available in the literature, can you identify any better compound, right, other than the one which reported in the literature, right. First we see that, okay, so we have to derive the features. You can see the different types of QSARs, maybe 1D descriptor, 2D descriptor and 3D descriptors, right, or the atom type, log P, molecular refractivity, right, different types of parameters we can derive, right. So, they discuss about the different methods like paddle or the V-life, right, or the power MV. These are the various software available. If we give your compound in spiral format or with the molecular formula or with the structure formula, it will give you all the data, right, it will provide all the data. So, here if you see these are the exponent values, right. So, then we can have the P1, P2 are the different property values, right. Then take the first property, I will tell you the partition coefficient, how to calculate this. So, here this is the X in aqueous region and this is octanol. This is the octanol aqueous partition coefficient. This is how we define the log P values, right, like the kind of hydrophobicity, right. The partition coefficient is the ratio of the concentration of compound in octanol to its water. This is any organic solvent, arcanol, ethanol we can use, right, and with respect to the water, this will give you the partition coefficient, right. How to calculate this? For example, here I give one compound, this is the benzene ring with the CH3 is attached here and the fluorine is attached here. So, how to calculate log P? You can get the values from the sum of the fragments, right. This is a parent compound, this is the parent compound, right, and you can see the other fragments, the CH3 or the fluorine and all. So, we see the benzene, this is a parent compound, this is 2.5, log P is 2.5 experimentally known, and the methyl group, this is the CH3, this group this is equal to 0.6 and the fluorine it is minus 0.4, right, and the Fi for the fluorine atom in the different conformation ortho to methyl group, this is the log P of the parent compound and this is the for individual fragments and this is the influence with these different groups. Parent compound is a benzene, so this is given as 2.5 and individual compounds CH3 plus F. So, we give the 0.6 plus 0.4 and because of the Fij this is minus 0.3, finally, we get the value of 2.4 in the case of this particular structure. Likewise, for any compounds you have, you can make as a core, the core we have the values, then all the constituents you can get the fragments, right, then the you can get the Fij values among these different groups and finally, to get the log P, the summation of all these values give you the log P for the particular compound. So, you can calculate. This is another example, get the molar refractivity. How to get the molar refractivity? So, it is a measure of the total polarizability of a molecule. So, you can use this equation MR, molar refractivity, this n square minus 1 divided by n square minus 2, right, multiplied by this molecular weight by density, this is the volume. This is the correction factor for the polarization. You can say n is the index of refraction, because refractive index is known for the compounds, and here this molecular weight by density you can give almost the volume. So, you give this compound, right, this is AICAR compound, right, this is amino imidazole 4 carboxamide ribonucleotide. So, you can say this n equal to 0.81 because it is index of refraction, it is known for all the compounds, we know the refractive index, right, this 0.81 square minus 1, right, then you can see the volume, this 258.47, right. So, you divide with this volume here, finally we get the value as 67.097, right. So, n we know that n equal to 0.81, so then we can use substitute the values and you can use the volume of the compound. So, finally you can get the MR equal to 67.097. Likewise, you can see any parameters, if you know the compound you can calculate. So, it is better to understand how to calculate, right, then you can use the software to get the derivative features, otherwise it is a kind of black box to get some numbers, right, numbers have no meaning. So, to understand how they get the numbers, at least you need to derive for some compounds and see whether this matches with the data which you get from the software, then we are sure that the values are high or low, why it is high and why it is low, depending on that you can interpret your Qsar equations, right. Now, we can have different values, right, molar refractivity, A log P and all the details. So, these are the various descriptors, right. So, here we have the ICVT values. Now, we can compare, right. For example, if you take only one, so this is one property, right, if you see that, then you can relate any property. Here I used one of these electronic properties, right, or the topological values. So, you can fit these experimental values using any of these properties, right, then use this equation, right, for any new compound you can predict the IC50 values. Here I show the values observed IC50 and the predicted IC50, right, some of them are having good relationship, like this is having a good agreement, and some cases it is not very good. For example, in this case, okay, this is not able to predict well because actual experimental is 3.7 and the predicted one is just 0.8, right. You can see several cases which are very close in this line, for example, if you put a line, right, this is close, but some of them are very far, right, these are very far, they are outliers, they are not able to, right, fit with the particular property. So, you can use any properties. So, each property can tell only the partial information, because the whole compound it is not depending upon any specific property, right. This is what we can observe from this one. So, only each properties they can capture some type of information, right. So, in this property at least you can get the deviation of 1.6, right, but some of the cases yes, but some cases no. In this case what can we do, right, you can combine different properties, because for example property P1 you have some information, right, and P2 can capture some other information in a particular compound. Then if you add up these two properties, right, then can better predict the activity, right, rather than using a single one. So, you can use different property values and see whether you can combine together to check whether this IC50 can be expressed in terms of the combination of different properties. Here I put P1, P2 and P3, three different properties, right. So, with some coefficients, right. If you see this equation, right, then here this is the observed and predicted IC50 values, most of them are very near to the line, right, this is the trend line, right. So, compared with this one, we can improve, right, very significantly when you combine few properties, right. Then you have to validate whether it is fine or not, you do with the jackknife test. In this case, if you have n data, n minus 1 are used for the training and the left out is used for the test. Do it up to n times and see how far is the correlation for the test set of data obtained with the jackknife test. So, if you see this one, okay, this is the training, this is very close, but if you go to the test set, some of them are close, but even then you can see several compounds which are far away from the line, right. That means, if you use all the data, it is able to fit, because the coefficients will fit the data, but if you take some of the one compound, right, for example, if one of the compound which are highly variable, then it is not able to fit, right. So, in this case we need to find what are the other the parameters which can also capture this particular compound and refine your model. So, in that case you can get the better equations. This is another example. So, this is the choline kinase inhibitors. Here what they did, they have the common core is common and there are different R groups. So, the various R groups for 39 compounds we have the IC50 values. How to do this? In this case they try to develop the force field based methods. For example, they use the electrostatic potentials 612 plus as the Coulomb potential, and from this one they are able to predict, right, the experimental activity with respect to the predicted activity with the high accuracy, right, in this type of inhibitors. Then explain another one important example, the BCL2 inhibitor. This is the work done in our lab. So, here the Y BCL2, this is stands for B cell lymphoma, right. This is also a cause for several types of cancers. So, we see in this graph, okay, here the different types of cancers, right, breast cancer, colorectal cancer or the prostate cancer and so on, and you can see the percentage of tumors which express the BCL2, right. This is very high. So, it is very important to identify some of the lead compounds, right, for as an inhibitors. So, this is a structure, this is mainly alpha helices, okay, you can see this is structure of this BCL2 and how to get these models. So, then if you look at the structure, the active sites, and you can see there are 7 core groups. So, these are 7 core groups A, B, C, D, E, F, G, different core groups, right. So, they belongs to the different cores like apocalypal or the quinolone and so on. So, based on that they find different types of compounds which show the activity, right. For example, the class A, right, there are several compounds, right, which have the activity, right, against these compounds, right. So, now we take the compounds, we have a set of compounds, for example 100 compounds or 50 compounds in each group, right. So, then we derive the equations, but if you combine all these groups together, it may not work fine because they have a different cores with the properties are different, but activities also different, right. In this case sometimes similar compounds different activities and the different compounds have dissimilar activities. So, it is very important to take the core and derive the equations. So, here I show example equations for the different types of families, right, and you have the different equations, right. Some of them you can see the properties are similar and some cases properties are totally different, right. In this case the compounds which describe any particular family depend upon any specific properties, right. So, here I show the results, right, for the different families. This is the number of data, right, it ranges from 27 to 89, right, and here number of descriptors, right. So, what is one of the rules in the QSAR is we should finally develop the model with less number of descriptors, go with the 800 700 descriptors. Finally, if we ended up with this less number of descriptors, here they use only 3 or 2 descriptors, right, and see the correlation, it is very high. For example, this is 0.9, 0.9 and so on, more than 0.9. This is the MAE, MAE is also very high, right. The last one is 5 because they give in percentage. So, in this case it is 5%, okay, this is the unit of the micromolar, right. So, in this case the MAE is also less and the correlation is also very high. Here the figure I show the predicted values, PIC50 is logarithmic IC50 values, right. So, the experimental values, right. So, you can see there is a good correlation between the predicted and the experimental IC50 values, right. You can see most of them are on the line, right. Now, if you do this, right, then you are able to assess the performance of method and this you can predict new compounds, right, and then you can see whether this could be a potential compounds or not. So, we did with some known drugs. So, these are the some of the FDA approved drugs, this one approved drug ABT 199 and the phase 1 clinical trial and preclinical trial. So, from different families, pyrosol family or apocalypal family and so on. So, these are the different inhibitors. So, we have these compounds, with these compounds we can get the properties, right, you can get the necessary properties here and use this equation to predict the IC50 values, right. This is the predicted IC50 values, right. So, this is experimental IC50 values. So, this is very close, this is 2 and 3, say 44 and 60 and 20 and 49, right, this is not a bad one. So, you can see the predicted values, right, agree well with the experimental values even for the known drugs, right. This is not used in the development of the model, so this works, right. So now we can predict the zinc. How many data in the zinc database? Around 35 million, 40 million sequences, right. There are various database and various number of compounds. So for each family or for each core, we can get the compounds in the zinc database or any other databases, right. The core should be the same, and you can have the different moieties, the chemical groups are different, right. So if you see, this is a zinc ID and you have the different families, right, you can see the IC50 values you can predict. So some of the cases you could see the very less IC50 values, in this case it could be a lead compounds. Then you can also do something more, if you take these compounds and play around the chemical groups, right. So we can change the chemical group and then again predict the IC50 values, and you can design your own compounds and see whether you can identify any new compounds with better activity, right. If you do that, then finally it is subject to experimental validations and see whether you could identify any new lead compounds, right, which could be a potential inhibitor for this particular target, right. So currently this we are trying to fuse with the new chemical groups, right, and we designed several compounds, and now we are doing these experiments to validate whether these new compounds, right, could behave better than the existing ones in the deposit in the different databases, right. So now we go, once we get these validations, get the inhibitory constants IC50 values, then go for the next steps and see whether we can get reliable results with the particular compounds, right, to the next levels, right, to be in the clinical trials, right. So what do we discuss today in summarizing?
QSAR.
It is a QSAR. What is QSAR? Quantitative structure activity relationship. So how to relate? So we relate the activity with respect to descriptors. What are different types of descriptors? So 1D QSAR at 2D, one is the molecular weight and get the some shape, size and so on, and we do the connectivity, or you can the 3D, we can go with this 3D structure information, right. So there is various descriptors, right, that we can have several databases to get the ligands. What are different databases available to get the information regarding ligands? Campbell, zinc database, right, Timbal, there are many databases, so you can get the data with reliable different types of information. Either you get the compounds or the structures or the activity, or you can get the pose. For example, if you take the PDB, so what is PDB? Protein data bank, right, you can see several identities, right, with the protein with ligands. So if you look into these ligands with the proteins, and you can see how the ligands interact with the proteins and how about the active sites and how about the interactions, right. So see any shape complementarity or the chemical complementarity between the hydrogen bond donor and acceptors, or the hydrophobicity and the pockets and so on. So you get the information, right. Based on that you can derive the descriptors, right, and then we have to do the feature selection. We do the subjective feature selection or the objective feature selection. How to do the objective feature selection?
Using correlation or
You can see the correlation, and if it is highly correlated, you can discard one, right. If subjective ones, we know some properties are important. For example, log P, hydrogen donors or acceptors. So you can keep this as a descriptor, right. You can do that. So what are the rules of the QSAR? Data set, okay, there is sufficient number of data compared with the descriptors, that you can at least more than 5 times, 10 times is serviceable, right. And then we need to, what are the other rules of the QSAR? So to combine the variability of this data, and there should not be the any repeated data sets, right. So we need to get all these rules. Then finally we ended up with the 3 to 5 important descriptors to define this any biological activity, right. So then we discussed about various types of validations and the model development, right. So then using that information you can develop a model, right, you can develop new models. So from that the molecular descriptors, right, can be explained, right, can be used to predict the biological activity, say IC50 values, right, and then you can derive this equation. Then you can use the new compounds, right, you can use these particular equations whether linear or non-linear, right, for the identifying the new compounds. So, once we get the new compound, then finally all the compounds are subject to experimental validations, right. In this case you can get the good results, then you can go for the next level of experiments, right, and go for these clinical trials and so on, right. So, till now we discussed about the bioinformatics aspects on the different protein sequences and protein structures and the folding stability interactions, as well as the structure based drug design and design the inhibitors, right. The next few classes I will discuss about the some sort of the programming, like the awk programming to kind of script to get the manipulate the PDB data or any data obtained in the database or from the programming different output of the programs. And also we see what are the various commonly available problems in the literature and how to deal with the problems, how to do with that one, and also see the statistical methods and machine learning techniques and few case studies to see how to systematically carry out in any project, right, and what will be the result and how to interpret the data and so on. Thanks for your kind attention.